As we all know, MAVs are revolutionizing our understanding of protein function and variant interpretation. Many groups have leveraged the fitness effect of disease-relevant gene knockout to perform dropout screens. This plot shows the distribution of the effect that gene knockout has on cell growth. Note that less than 25% of genes, both disease and non-disease relevant, are essential in cell culture. So while we are resolving unknowns in some clinically relevant genes like those featured here, most disease-associated genes, including MUTYH, will need non-cell fitness assays for variant analysis. MUTYH is involved in oxidative damage repair. Atoxyguanine is the most common form of oxidative DNA damage. It can base pair with cytosine or be mispaired with adenine upon DNA replication. Without repair, this mismatch can become permanent and results in GC to TA transversion. MUTYH recognizes this ATO-G to A mismatch and excises the adenine, initiating long patch BER. Then OG1 and short patch BER restore the original GC pair. Loss of MUTYH has consequences on multiple levels. Offspring of a MUTYH parent with two pathogenic variants have a modest increase in a C to A mutation frequency, which is seen in this family with MAT variants and has been shown in MUTYH variants and has been shown in several other studies. A similar C to A accumulation is seen in somatic cells without MUTYH, which is nearly the same mutation signature as that of ROS, of reactive oxygen species exposure. Related to the colon, this is related to the colon's high cellular replacement and respiration rates. Colon cells are especially prone to C to A mutations, which, when these occur in APC or KRAS, can lead to cancer. This MUTYH-associated disease is an autosomal recessive polyposis with early onset colorectal cancer. While the prevalence is rare, the rate of carriers is high, nearly 2% for European ancestry. Unfortunately, many people inherit one pathogenic founder allele and one variant of unknown significance, or VUS. And as shown in this chart, most VUS are missense variants, which are difficult to interpret. Thus, focusing on these missense VUS has the potential for high clinical impact. To test MUTYH function, we generated a fluorescent reporter with an ATO-G to A mispair in GFP using a PCR-based method to generate single-stranded DNA and incorporate ATO-G opposite adenine. You can see successful ATO-G incorporation via Sanger sequencing with this spike showing stalling right before the ATO-G, and then either A or C is the following base pair. So how does this reporter work? There is an ATO-G to A mispair in an early stop codon, and RNA is transcribed from copying this bottom strand. Thus, when RNA is translated, it results in a truncated GFP and no fluorescence. If MUTYH excises this adenine, the normal glutamate codon is restored and GFP is expressed. We transfected cells with this reporter in an M-Cherry control and measured MUTYH function by flow cytometry. All cells on the top of the plot express M-Cherry and are the transfected portion. Cells in the top right also express GFP, representing functional MUTYH. Then comparing the wild type to knockout cells have four-fold lower GFP signal, representing less repair. We added variants of known function to knockout cells and measured reporter repair. These variants have been extensively studied and their functional impact is well established. The polymorphism V8M has the same repair capacity as wild type, while the partial loss of function G368D has 50% repair, matching with its lower penetrance clinically, and the complete loss of function Y151C is comparable to knockout cell repair. We next set out to apply this reporter assay in a pooled fashion in order to measure the functional status of all MUTYH missense mutations. We generated a library of the over 10,000 single codon mutations and used lentiviral constructs to stably introduce these into knockout cells, so that each individual cell expresses a different MUTYH mutation. Then we used our AtoG reporter and cell sorting to separate cells by MUTYH status into repair positive and repair negative pools. Based on variant frequency in these repair pools, we calculated a function score, for which we see perfect separation of nonsense in pink and synonymous in green variant function scores. Missense variants span a range of function scores, most of which are wild-type-like. Overall, we see strong concordance with mutation fluctuation assays, in which mutation frequency is measured by growth of bacteria expressing MUTYH variants. Variants with lower function have a higher mutation frequency. Thus, we see concordance with loss of function variants in our assay having a high mutation rate in bacteria. Here we show the results in a variant effect map, where along the x-axis we have all the codons of MUTYH, and the y-axis is the amino acid substitutions. Dark red boxes represent loss of function variants, orange are intermediate function, and white are wild-type-like. Early stop codons are deleterious, as well as most proline substitutions in secondary structures, while this unstructured N-terminus is tolerant for all missense variants. And the C-terminal region is tolerant to all substitutions, including truncating variants. Thus, the C-terminus, including PCA binding at this pit box, is dispensable for our assay. The adenine excision catalytic region of MUTYH is generally intolerant to substitution, while the AtoG recognition domain is more tolerant to mutations. And then when we look at expanding our view into population databases, we see that pathogenic MUTYH variants are routinely found in them, which is consistent with MAP's recessive inheritance. Here we show function scores in our assay versus the population frequency. That is the sum of the NOMAD, non-UK biobank, and RGC allele counts, separated by variant class. Larger points correspond to variants that are observed in the homozygous state, which includes several pathogenic variants. The founder alleles make up half of all rare missense alleles in NOMAD, that is, having a minor allele frequency less than 1%. But looking at all the other 169 abnormal variants, which are individually rare, but together have a cumulative minor allele frequency that is comparable to Y151C. We then wanted to explore how our data could be used to improve variant interpretation agnostic to genetic ancestry groups. To do this, we plotted missense single nucleotide variants by NOMAD allele frequency, with each variant represented by a line connecting the allele frequency in primary European ancestry group to the allele frequency in the non-European ancestry. We identified 318 missense SNVs with at least 5-fold higher frequency in the non-European ancestry groups, which are seen in this plot as the tall arrows. As expected, most of these variants are neutral, and about 25% are intermediate or loss of function, of which the great majority are either absent from ClinVar or they have VUS or conflicting interpretations, despite being on average 220-fold more common in their respective ancestry groups. As for applying our functional assay findings to ClinVar, we have perfect separation of variants with known function, providing the opportunity to use our assay to reclassify variants with unknown or conflicting reports. To do this, we calculated odds-path scores, which is a quantitative framework for classifying variants following ACMG guidelines for a gold standard set of MUTYH benign and pathogenic variants reported in ClinVar. We performed stringent curation to first test variants with confident classifications, that is, only include the variants with multiple submissions for polyposis, and we found perfect agreement of pathogenic and benign classification with our assay's findings. Relaxing this filtering to include more variants, such as those with conflicting reports that we classified based on majority reported classification, we see near perfect concordance, and actually 2 of the 3 discordant variants are in the unstructured C-terminal domain, which we have noted as non-essential in our assay. Not only were we lucky in that we have many ClinVar classifications to test against our assay, we also have a large set of classified missense variants with individual level clinical data, which again showed near perfect concordance. Note that for all sets, strong classification level is reached by our assay. Thus, our functional assay can be applied to VUS reanalysis. In fact, Ambry Genetics recently shared that our assay is being used to aid in reclassification of variants found in over 5,000 patients. With our assay and clinical data from Ambry Genetics, we are able to test disease risk based on variant function in a large dataset of nearly 400 individuals carrying 2 MUTYH variants. One known pathogenic variant and another variant which is either pathogenic, benign, or VUS. We then group these individuals based on the other variants, as in the non-known pathogenic variant, this other variant's function, as whether it's neutral, intermediate, or loss of function. Here you can see in gray that people heterozygous for a pathogenic and a neutral allele have no increase in disease risk, while homozygosity of the intermediate G368D allele carries significantly increased risk. But this is still less penetrant than the loss of function Y151C allele, matching with previous biochemical, clinical, and population analysis studies. Further, when we look at inheritance of a pathogenic allele with an intermediate variant, they exhibit a range of lower penetrance than G368D, which has a pathogenic intermediate function. While inheritance of a pathogenic allele and a loss of function variant exhibits similar disease risk as inheritance of one Y151C and one G368D allele. Looking at age of onset, unaffected individuals with a known pathogenic and neutral variant are in gray here and confer a population level risk for non-polyposis cancer, colorectal cancer, or other tissue. We observe a mild shift in MAP age of onset for individuals with a pathogenic and an intermediate variant. And we see a significant lower age of onset for MAP disease onset for individuals with pathogenic and loss of function variants. Then when we look at average function scores per residue, we see overall mutations in buried residues are more damaging than in residues on the outside of the protein. As for this residue in the unstructured domain, completely intolerant to substitution. And it's the first amino acid. So only methionine is tolerated. That's what we expect to see. But these other two residues are interesting since they have a strong negative function score despite being exposed residues, which are generally more tolerant to substitution, which is seen in the clustering of the other exposed residues in the neutral and intermediate average scores. These residues have clinical reports of pathogenic variants and other reports of pathogenic variants and other functional assay studies characterizing loss of function variants at these residues. Thus, these findings are unlikely to be errors in our assay. It is possible that these are critical for APE1 binding, which interacts with the A basic site left after MUTYH excises adenine. Looking at these residues in the 3D structure colored by function score, we can see that they are adjacent to other constrained residues. And they are both exposed near each other, so an interaction with APE1 is possible. Since MUTYH is a DNA binding protein, we want to see if there's a selection against negatively charged amino acid, that is D or E substitutions. So we compiled all residues within 5 angstroms of DNA shown in this heat map and compared the intolerance scores of D and E variants, both near and not near DNA. Since the DE intolerance scores are more negative for residues that are close to the DNA backbone, this is suggestive that electrostatic repulsion is the cause of intolerance for DE substitutions near DNA. The common founder allele G368D is one such negative substitution near DNA, expected to have both steric and electrostatic hindrance. Due to MUTYH's sensitivity to expression levels affecting pathology and genome integrity, half of the non-neutral alleles fall into intermediate function category, and there's incomplete penetrance with later age of onset for intermediate variants. We aim to further study how expression of MUTYH affects function. We compared variant function scores from the reporter assay performed with endogenous expression levels to 100 times that. Looking at G368, we see an overall shift away from negative loss of function and intermediate scores to orange and white, which are intermediate and wild-type-like scores. Overall, at this whole section, we see a shift towards higher function scores, not just at that residue. That is, we see overall less loss of function with overexpression. Note that the intermediate variants that are neutral with overexpression have low average fold X scores. That is, they're predicted to be more stable. And another thing to note is prolines and secondary structures, such as in this beta sheet, disrupt stabilizing hydrogen bonds between the amino acid backbones. So they exhibit only a mild increase in function score with overexpression. To recap, our variant to function map covers over 97% of all possible MUTYH point variants, and we achieved 100% accuracy in classifying the pathogenicity of these known clinical variants. And we were able to recapitulate known functional differences, and we see that more severely impaired missense variants confer a greater disease risk. Going forward, we aim to investigate some of those sites that I noted of complete missense intolerance. And also, our map is already being used as a resource to address the over 1,100 unresolved variants, over 300 of which are reasonably common in specific population groups. And of course, I would like to thank my lab for all of their help, as well as our funding sources to make this all possible.
Thank you, Shelby, for the great presentation. If anybody has questions, please put in the Q&A, and I can read it. We have one question so far from Charlie Rowlands. Hi, Shelby, amazing work. Although UK Biobank doesn't have as many compound heterozygotes, homozygotes, as the clinical data set, have you still tried conducting odds ratio, um, uh, age of onset analysis using UK Biobank, given the likely inflation of effect size in your clinical data set due to ascertainment bias?
We have not. I think we're in the process of looking at UK Biobank data, but I don't have any of that analysis done. But we definitely aim to perform this. And yeah, that's a great question, and we would like to.
Okay, great. So I have a question. I think in your conclusion, you said your approach covered like 97, sorry, percentage of the number of variants in the gene. And I was wondering if you could like comment on the limitations of your approach. That would be like main explain this 3% that you didn't
Oh,
cover.
yes. Yes. So we did create all potentially possible, all these possible mutations, except some of them, if they created a restriction enzyme site that was part of our cloning to generate these plasmids, then that mutation wouldn't have been included. And but that was like less than 1% of variants, but the other variants that didn't make it into the analysis were ones that were had a very low copy number that we excluded from analysis due to you couldn't really analyze something that was too lowly represented, where any effect could have just been by random chance.